If you are not feeling great about the idea of running into one of these on your exam, this video is for you. Hi, I'm Sarah, and I'm your other biology professor, and let's work some problems with codon tables. First off, you're going to need your own copy of a codon table, or codon chart, or dictionary, or codon wheel, whatever you call it. If you know which one you'll have access to on your exam, that's what you should use for practice every single time. You'll get familiar with it and you'll be able to work faster with it. I'll leave this one up when we're working problems in this presentation. They all have the same kinds of information, so you're going to input codons written 5' to 3', and you're going to get out amino acids, or a signal that translation's done. Here we've got the codon GUA, read 5' to 3', just like how you're seeing them laid out in our codon table. Let's go over to that table and see what amino acid that codon's going to give us. So first base G, second base U, so that means we're working in this box down here. Third base A, and that's going to give us the amino acid valine. This means that when we're in the ribosome and we are translating from an mRNA, and that mRNA has the codon GUA, a tRNA will come in with a complementary anticodon, and that tRNA is going to be charged with valine. What would be the sequence on that anticodon? Remember your base pairing rules for RNA. G pairs with C, U, A, U. All right, I've got a bunch of codons for you now, a little mRNA strand. See if you can translate this one. Pause the video and come back when you're done to check your work. Ready? All right, if you didn't get something that looks like this, stick around and I'll walk you through it. But if you're feeling good, skip ahead. I've got one more problem for you. Just like the ribosome, we're going to work from the 5' end towards the 3' end, looking for our start codon AUG. I put it right on the end in this one. You may have a little more work to do on sequences that you get on your exam. AUG is our start codon. It's labeled right here as start. It's not always labeled in all tables, but that's going to be the first one that we add. The next codon is UUU, and if we go to the chart, you can see that's phenylalanine. Next one, CUU. CUU gives us leucine. And you'll notice that I'm drawing these lines to mark my codons. If you're able to write on your test paper, I would recommend you do the same thing. It just helps you keep track of your codons. So the next one is UAC. UAC right here is tyrosine. And then AGU, serine right here. And then UGA, which we can see right here, is a stop. So that means that we are done. This is the end of our little peptide chain. We don't have tRNAs with anticodons complementary to stop codons. Instead, there are other proteins called release factors. And these release factors are going to come in, they're going to bind to the stop codons, and they're going to basically blow up your translational machinery and release our little peptide, let it go do its job. And one final note here, just like how our nucleic acids have directionality, 5' and 3' ends, our peptides are going to have directionality as well, N-terminus and C-terminus. The N-terminus or amino terminus is the first side that gets made. It's where the methionine is going to be. And the C-terminus is where we're always going to add the next one. And it's going to be at the end of our chain, in this case over by serine. Let's try one more on your own. And I'll drop a codon table up here just in case you've been using mine. So take a second, pause the video, work through it, And when you're ready, hit play and I'll show you my solution. Did you get it? You should have a peptide with 6 amino acids, starting with methionine, and I've got the rest of them labeled on here. I wanted to point out, you can see the 5th amino acid here is another methionine. That's a place that a lot of students get confused. Methionine is always the first amino acid, but it can also come later on in your peptide. So just a quirk that I wanted to make sure you saw now instead of on your exam. Keep practicing. Remember all those tips we covered, and you'll be in great shape. And if you want even more practice, I got you. Be sure to like the video if you found it helpful, and check out my website for even more biology content. And I'll see you next time.